Bismillahirrahmanirrahim. Today I am going to tell you about the choroid plexus and ependymal cells. Choroid plexus and ependymal cell both play a very key role in our brain and also connects with other parts of the body. First of all, I am going to tell you about the choroid plexus. Choroid plexus is a plexus of cells that arise from the choroidea in each of ventricles of brain. Mainly in each ventricles play a very key role in our human brain. The function now I'm going to tell you about the function of choroid plexus. The first function of choroid plexus is to produce the most important function is to produce the CSF, cerebrospinal fluid. Second one, this cerebrospinal play a very important role in our brain. It reduces the friction of the brain and much more functions also. Second one, CSF produced and secreted by regions of choroid plexus. And third one, choroid plexus resides the innermost layer of meninges, or I can say that pia mater. So it's the role of choroid plexus is very important. If any pathology is occur in the choroid plexus, we face a lot of disturbance in our brain. The most important pathology is neoplasm. Pathology is occur in the choroid plexus. We face a lot of disturbance in our brain. The most important pathology is neoplasm. It is a tumor in the brain. And I'm gonna tell you now about the ependymal cells. Ependymal cells also play a very good role in our brain. First of all, ependymal cells are also known as epithelial cells that line the ventricles of brain. In our brain, ventricles are present. Ependymal cells are lined by these cells. Second one is they also take part in good role in fluid homeostasis. I mean that ependymal cells play part in fluid maintenance and also in blood-brain barrier. If any pathology occur in these cells, we face a lot of disturbance in our brain also. Thank you.
Today I am going to explain venous sinus thrombosis. Venous sinus thrombosis कब होता है? जब किसी भी साइनस में क्लॉट बन जाता है या कोई थ्रोम्बोसिस हो जाता है। और ये होता क्यों है? ये हाइपरकोगुलेशन की वजह से होता है। हाइपरकोगुलेशन के ज़्यादा चांसेस कब होते हैं? जब प्रेगनेंसी के दौरान ओरल कॉन्ट्रासेप्टिव पिल्स के दौरान और इसकी सिम्टम्स में आता है वेनस साइनस थ्रोम्बोसिस की में हेडेक, डिप्लोपिया, डबल विजन एंड फटीग, muscle weakness, etc.
Explain the topography of lateral third and fourth ventricle. CSF is found lateral ventricle and enter third ventricle by passing through foramen Monroe. From here it passes fourth ventricle through the aqueduct Salvius. From fourth ventricle, CSF entered through cisterna magna and cisterna lateralis through the foramen magendi and foramen luschka. Foramen magendi is the central part and foramen luschka is the lateral. Cisterna magna and cisterna lateralis, cisterna magna and cisterna lateralis through the CSF circulate through the suborachnoid space over the spinal cord and cerebral hemisphere.
Assalamu alaikum. Today we will discuss about biochemistry और composition of cerebrospinal fluid. आज हम cerebrospinal fluid की composition और biochemistry के बारे में बात करेंगे। जो cerebrospinal fluid है वो दो मेन कंपोनेंट्स पर मुश्तमिल होता है, वाटर और सॉलिड्स। इसमें वाटर की रेशियो होती है 99.13% और उसके बाद जो सॉलिड्स होते हैं उनकी रेशियो होती है 0.87% रेशियो सॉलिड्स की होती है। इनमें से जो सॉलिड्स होते हैं वो आगे मज़ीद दो कंपोनेंट्स पर मुश्तमिल होते हैं, ऑर्गेनिक सब्सटेंसेस और इनऑर्गेनिक सब्सटेंसेस। ऑर्गेनिक सब्सटेंसेस में जो होते हैं प्रोटीन, अमाइनो एसिड, लैक्टिक एसिड, यूरिया, यूरिक एसिड, उसके बाद शुगर, कोलेस्ट्रॉल वगैरह वगैरह। और उसके बाद और इनऑर्गेनिक सब्सटेंसेस में आ जाते हैं सोडियम, कैल्शियम, मैग्नीशियम, पोटाशियम, क्लोराइड और फॉस्फेट।
My topic is
lumbar puncture. Lumbar puncture with fluid collections may also be a part of other producers such as an X-ray or CT scan after dye has been inserted into CSF. Rarely other methods of CSF collections may be used. External puncture uses a needle placed below the occipital bone back of the skull. It can be dangerous because it is so close of brain stem. It is always done with fluoroscopy.
Today I am going to explain the dural venous sinuses, which are also known as cranial sinuses, dural sinuses, or cerebral sinuses, and its drainage. The dural venous sinuses are the venous channels which are present between the endosteal and meningeal layer of the dura mater in the brain, and these get drainage from the cerebral veins and also the CSF drainage from the subarachnoid space via arachnoid villi and granulations. These dural venous sinuses consist of few paired and few unpaired venous sinuses. Paired sinuses include the transverse sinus, sigmoidal sinus, cavernous sinus, superior and inferior petrosal sinus, and sphenoparietal sinuses. While the unpaired dural venous sinuses include the superior sagittal sinus, inferior sagittal sinus, and straight sinus. Now I will talk about its drainage. The superior sagittal sinus, which also forms the depression in our cranial bone on its inner surface, and inferior sagittal sinus and superior sagittal sinus are separated by the falx cerebri, which is a dural fold which is present between the two hemispheres. The inferior sagittal sinus get drainage also from the great vein of the Galen, which is also called as great cerebral vein, and the great cerebral vein also get drainage from the basal vein of the Rosenthum. And at the junction of the falx cerebri and tentorium cerebelli, the inferior sagittal sinus and the great vein of the Galen forms the straight sinus. The superior sagittal sinus, the straight sinus, and the transverse and the occipital sinus are drained into the confluence of sinus, and the confluence of the sinus give two drainage, which are called as transverse sinuses. The transverse sinuses also form the depression in our cranium bone on its inner surface. The transverse sinus gets also drainage from the superior petrosal sinus. The cavernous, as we know, the cavernous sinus, which are present on the lateral side of the sella turcica, and its get drainage from the sphenoparietal sinus, and also the superior orbital vein drain into the cavernous sinus. The cavernous sinus give two type of the drainage, which are called as superior petrosal sinus and inferior petrosal sinus. Superior petrosal sinus drain into the transverse sinus, which becomes the sigmoid sinus after passing through the tentorium cerebelli. The sigmoid sinus are the paired sinus of our brain, which are S-shaped, and after passing from the foramen magnum and to the lateral surface, after passing from the foramen magnum to its lateral surface, which has also a foramen which called as jugular foramen. After passing it becomes the internal jugular vein, and internal jugular vein gets drainage also from the inferior petrosal sinus. The internal jugular vein then with external jugular vein, subclavian vein becomes the brachiocephalic vein on our both sides, and then it becomes it's drained into the superior vena cava, which eventually drain into the right atrium of the heart. Thank you.
Today my topic is the formation of blood-brain barrier. So let's start. Blood-brain barrier is a neuroprotective structure that prevents the entry of many substances and pathogens into the brain tissues from blood. It was observed more than 50 years ago when trypan blue, the acidic dye, was injected into the living animal. All the tissues of the brain were strained by it except the brain and spinal cord. This observation suggested that there was a hypothetical barrier which prevents the diffusion of dripping blue into the brain tissue from the capillaries. This barrier was named as blood-brain barrier. It exists in capillary membrane in all parts of the brain except in some areas of the hypothalamus. Structures of blood-brain barrier. Tight junction in the endothelial cells of brain capillaries are responsible for blood-brain barrier mechanism. Functions of blood-brain barrier. Blood-brain barrier act as both mechanical barrier and transport mechanism. It prevents potentially harmful chemical substance and permit metabolic and essential material into the brain tissue. Blood-brain barrier provide healthy environment for the nerve cells of the brain. Substance which can pass through blood-brain barrier: oxygen, carbon dioxide, water, glucose, amino acid, electrolytes, drugs such as L-dopa, 5-hydroxytryptamine, sulfonamide, tetracycline, etc. So lipid-soluble gases such as ether and nitrous oxides and other lipid-soluble substances. Substance which cannot pass through blood-brain barrier. First, injurious chemical agents, pathogens such as bacteria, drugs such as penicillin and the catecholamines. Dopamine cannot pass through blood-brain barrier, so Parkinsonism is treated with L-dopa instead of dopamine.
Alaikum wa rahmatullahi wa barakatuh. Today I will talk about function of cerebrospinal fluid. Number one, protective function. Protective function, CSF acts as fluid buffer and protects the brain from shock. Since the specific gravity of brain and CSF is more or less same, brain fluids in CSF when head receives a blow, CSF acts like a cushion and prevents the movement of brain against the skull bone, and therapy prevents the damage of brain. Number two, regulation of cerebral content volume. Regulation of cerebral content volume is essential because the brain may be affected if the volume of cerebral content increased. It happens in cerebral hemorrhage and brain terms. Number three, medium of exchange. Medium of exchange, CSF is the medium through which many substances, particularly nutritive substances and waste materials, are exchanged between blood and brain tissue.
Shunt in the XQ of the hydrocephalus, the CSF is not properly reabsorption and followed with the dilation of ventricles. The treatment for this type of hydrocephalus is peritoneal shunt. In this process of the peritoneal shunt, the pathway of CSF is connected to the peritoneum to reduce the intracranial pressure. And the CSF is diverted from the lumbar thecal sac to the peritoneal cavity.